Hello, my name is Stephanie Angel, and I'm currently an undergraduate senior at the University of Toledo, majoring in biology and minoring in chemistry with a pre-med concentration. I conduct my research under the mentorship of Dr. Vestal, here at the University of Toledo Medical Campus. Cancer has become one of the most prevailing diseases in our society today. With more than 1 million new cases diagnosed every year, chances are you know somebody with this disease. Thanks to our scientists, many new information and treatments have been used in the battle against cancer. For my research, I explored a specific type of cancer called glioblastoma. Glioblastoma is a stage 4 brain cancer that contains tumors that arise from supporting tissues of the brain. Life expectancy for patients, even with aggressive treatment, is about 14 to 16 months. With that in mind, my goal is to help expand the knowledge available about glioblastoma. Jacob Justinger, a previous member of our lab, had actually found that the protein GTPase is elevated in glioblastomas resistant to temozolomide. Temozolomide is a commonly used chemotherapeutic drug against glioblastomas because of its capability to penetrate the blood-brain barrier. GTPase is a protein already known to increase the evasiveness of glioblastomas, and it is known to be involved in drug resistance of another chemotherapeutic drug called paclitaxel. For my research, I wanted to explore the possible involvement of GTPase in temozolomide resistance of glioblastomas. In order to do this, I asked 2 questions. My first question asked, is GTPase alone sufficient to cause temozolomide resistance in glioblastomas? In order to do this, I artificially elevated the levels of GTPase in glioblastomas sensitive to TMC. I then performed a colony forming assay in order to observe cell growth when exposed to this drug. I also asked a second question. Is GTPase required for temozolomide resistance? In order to answer this question, I plan to knock down the levels of GTPase in glioblastomas already resistant to the drug temozolomide. I'll then perform another colony forming assay in order to observe the cell growth when exposed to this drug. Temozolomide is also known to function by causing autophagy in glioblastoma. Autophagy is commonly used in cells in order to degrade any unnecessary or dysfunctional cellular components, but it's also known to cause cell death. I also wanted to explore the possible association of GTPase with autophagosomal membranes. In order to do this, I used immunofluorescence. This type of procedure places a fluorescent tag onto a desired protein. Then this makes it capable for you to image the distribution of the protein in a cell. By performing these experiments, I hope to be able to answer the questions pertaining to GTPase's involvement in glioblastoma. Therefore, I'll be able to expand the knowledge provided by researchers around the world about glioblastoma. For these past few months, I have enjoyed myself working with all these cells and by finding results using the scientific process. I hope that as you look at my research, you find yourself enjoying it just as much as I had. And feel free to ask any questions. Thank you.